Hi, guys. So I wanted to talk to you about the sliding filament theory. And so hopefully they don't come on and make announcements while I'm doing this, but they've been making a couple, so there is a chance. Just want you to be aware of that. So I'm going to start right here. I've put together a little PowerPoint piece. With the anatomy of the muscle, because we need to look at how a muscle is built. And I always think of a muscle kind of a string cheese. So, you have the big piece that's the string cheese, but then you start untwisting it and you pull off smaller bundles, and then you can actually separate the smaller bundles into individual strings. And so we find that skeletal muscle actually is put together in a very similar way. So each muscle is composed of bundles of muscle fibers. And then these bundles will be in smaller groups and smaller groups. So each muscle fiber, so we have one bundle, we call, we have a bundle of muscle fibers and bundle muscle fibers, a whole bunch of bundles of muscle fibers, that makes the muscle. Now we're going to look at just one bundle. So a muscle fiber has multiple nuclei, and it's a cluster of what we call myofibrils. Myofibrils have two key types of proteins that basically have an overlapping pattern. This is what causes the striations on the skeletal muscle. So one of those is myosin. Myosin is the thicker filament, and actin is a thinner filament. So here, the muscle, you can see how we have all of these little individual bundles. And then when we pull out and look at one bundle in particular, we can get a single muscle fiber. So we have our muscle and our bundles, and we pull out one of the bundles. And notice that that bundle still is made up of individual ones. Those are the fibers. And then we can actually pull out, because really and honestly, the muscle fiber is still a little bundle, but this bundle we pull out is called myofibrils. That's what we were just talking about. And the myofibrils are separated into, we have like sections that we call sarcomeres. That's a banding pattern, but what it's made up of are these little teeny tiny myofilaments, which are our actin and our myosin. And so if you look, you see we have thicker ones and we have thinner ones. And remember, the thicker ones are the myosin and the thinner ones are the actin. And how these line up is what actually makes our banding pattern and our sarcomere. And that's what you're going to be working with when you're creating your project. So a sarcomere is kind of the key functional unit for contraction in the skeletal muscle. So this is the area where the contraction and relaxation is going to occur. So we locate it between what we call Z lines. We have two Z lines. Those are like the boundaries of the sarcomere. And one end of each of those Z line, we attach the actin filaments to an end of the Z line. The myosin filaments are kind of lined up in the middle between each of the actin filaments, and so they overlap. Like I said, this is ultimately what makes that banding pattern for the striated muscle. So a few terms that you might want to know. We have our epimysin, which is the outer covering of the whole muscle. The fascicle is one of the bundles of muscle fibers. So epimysin all the way around. Fascicle is the wrapping around one of the bundles. Then we have our perimysin. So each of these fascicles has a perimysin around it. It's like the outer wrapping. And then we have endomysin, is the thin covering around each muscle fiber. So the individual fibers. Both the perimysin and endomysium have blood vessels, nerve endings to make sure that the muscle tissue is getting everything that it might need. And then the myofibrils, remember that's an individual muscle fiber or muscle cell, and it has a bunch of the little sections. So we've got our muscle fiber that we pulled out, and now we can pull out the next bundle, which is the myofibril, right? Okay. And that myofibril is our two proteins. We have our thin filaments of actin and our thick filaments of myosin. I know this is repeating a little bit, but it's really important to understand that so we can move into the next unit, because the sarcomere is where the sliding filament and the contraction actually occurs. So we've got to understand the structure to see where we get. So muscle breaks in parts, until here we're looking at a sarcomere. And this black and white one is actually like a real X-ray kind of photograph. And then we have an artistic rendering. And remember, we said that the actin is attached to the Z bands. And then the myosin is kind of lined up in the middle between each of the actin sets. Okay. So you need to know the I bands are kind of this open space between strips of myosin. So we notice we have an I band on each end. The A band is the darker part shown by the presence of the myosin, those thick bands. The M line is kind of that central middle point, and so you can see it really does show up a little bit darker. The H zone is the space where the actin is not typically present. And all of those stripes pieces together make up the sarcomere. There is a coloring one that specifically helps you with, that's one of your choices, with the sarcomere in and of itself. And then there's also a coloring one that's going to talk about the sliding filament theory. Those might be two good choices to do in preparation for your project, as well as, of course, getting your notes from this. And so this is just another view of the same thing. So we've got the sarcomere and we've got all of these different lines and bands. Yes, you do need to know these. All of these things have to be labeled on your project product. So the sarcomere is what actually gives the muscles its striations, and they come from how these different bands line up and the overlaps versus non-overlaps. That's what actually gives the various colorations and banding pattern. The A band is going to be where the actin and myosin overlap. The I band is an area that only has actin. The Z lines, where the I band attaches to the actin, and the Z lines are also the boundaries of the sarcomere. So when we look at the Z bands, they're going to be between the two, that's the sarcomere goes between the two Z lines. On the other side of this Z line would be a whole nother sarcomere. So one sarcomere on one side, one sarcomere on the other. Those are like little dividing between sarcomeres. So that takes us specifically to the sliding filament theory. So the idea is that the myosin filaments will pull the actin. So if here's the myosin, it's going to grab and pull the actin over. So the myosin filaments pull the actin filaments towards the center of the sarcomere. This causes the sarcomere to shorten, which in turn shortens the muscle. So it starts with a neural impulse sent to the muscle that says you want to move. So we've got a little nerve signal that comes in and says, okay, this is what we want to do. The neuron releases a neurotransmitter called acetylcholine over the surface of the muscle, and the neurotransmitter then enters the muscle. So that neurotransmitter entering is basically what's going to tell the muscle, this is what to do. That neurotransmitter causes these sacs to release calcium. Calcium is kind of the start point of a contraction. The calcium triggers the binding sites on myosin. They become uncovered. When they're uncovered, the actin fibers can attach and move up the binding site so the Z lines get closer. So it really is like it's little hands that keep grabbing and pulling. The calcium is then reabsorbed, so those binding sites get recapped, so they're covered up, they can't grab anymore, and that's what allows for the relaxation of the muscle. So the idea here is anything that is shown in pink on the diagram is actin, anything that's shown in blue is myosin. So the myosin pulls the actin towards the center of each sarcomere. So we're pulling towards that M band, that was an M line that was in the middle. That's what causes it to shorten and contract. But understand that this is an all or nothing response. So the shortening of the sarcomere will occur along the entire length of the muscle fiber. It's not just going to do a little section, it's got to do the whole thing. The strength of that contraction, how strong that contraction is going to depend on how often the little individual fibers are stimulated to contract. The more stimulation, the stronger the contraction. How many muscle fibers contract within a given muscle. So remember, it's like we have strings wrapped around strings, wrapped around strings. We've got all these little bundles. And so if we only do 1 or 2 fibers, that's not going to be as strong a contraction as if we did 54 fibers, right? The more fibers involved, the stronger it will be. So this is what we're talking about with skeletal muscle contraction. So we get the signal from the nerves. And so this is just kind of showing you the nerve connection. So it is controlled voluntarily by the nervous system. And we use what we call a motor unit or motor neuron. And all of the muscle fibers that it controls are all going to be involved in the contraction. So that neuron, every single fiber that's attached to the same neuron, when it sends the signal, they're all going to contract. So hopefully that helps you out a little bit. If you have questions, please ask, right? Okay, so I'm going to stop sharing this specific screen. And I have one other picture I want to pull up and show you. So if I were working on the project, I would, according to the project, you have to have 2 images. You have to have the sarcomere in a relaxed state. And so you'll notice in this first or top picture that we are in a relaxed state, meaning everything is kind of spread out as far as it can. We've got our spaces for the Z line. We've got our spaces for the M line. We have that area where we're not having the overlap or the actin and myosin aren't as tight. And we've got each of the different items identified. So really and honestly, I mean, this is kind of the picture you need to draw. You just have to add your explanations for everything as well as having the labels. And you have to make sure the labels are on both diagrams. And then the bottom B, this would be a contracted sarcomere. Notice that our actin and myosin are pulled and overlapping almost everywhere. They're really tight. We don't have a lot of the empty spaces. Okay, so hopefully that will help you with the project as well. And like I said, if you have any questions, please ask. Bye.